Welcome back class 11 to the online bio classes. We were already doing chapter 5 morphology and anatomy of flowering plants, in which I have already discussed in that interactive classes regarding morphology is the study of external form and structure of an organism. If we study the plant morphology, means we are dealing with the external features of the plant like root system or the shoot system. But once we study the internal structure of root, stem or leaf, then it is called as anatomy, study of the internal structure of different plant organs, like what is the anatomy of stems of monocot and dicot, how they are different from each other, how the leaves are made of which types of tissues like mesophyll tissue, how many types of mesophyll tissues are there. Mesophyll tissues are differentiated into two main types, that is palisade and spongy tissue. So palisade are columnar and spongy is circular or oval in shape. Then we have to study about the secondary growth in dicot plants. You know that monocot plants generally do not grow in thickness due to absence of secondary growth, but in dicot plants they have secondary growth, which results in the formation of thick stems and roots. Then today we will discuss about what is vascular bundles. Already I said vascular bundle means the one which consists of xylem and phloem. Like if this part is xylem, the upper part, then the middle one, this layer of cells, it is called cambium, and cambium is made up of actively dividing cells like meristematic cells. It results in the formation of secondary xylem and secondary phloem. Then the lower one we can take it as phloem. Together this structure, in which xylem and phloem both are called the conducting tissue. Conducting tissue, you know, or vascular tissue, are the one which helps in the conduction of water and minerals along with the food molecules. Like water and minerals are conducted by xylem tissue. The food molecules are translocated from the leaves to all parts of the plant through the phloem tissue. Similarly, from all parts of the plants, the food can be translocated through the phloem tissue. So this is a diagram of one vascular bundle. The arrangement of vascular bundles in monocot and dicot we studied was different. One of them is arranged in the form of a ring-like structure. I will just give you a review of this chapter. In anatomy of flowering plants, we studied about the tissue. You know the definition of tissue is a group of actively dividing cells that perform a particular function. Plant tissues are generally of two types. What are they? Meristematic tissue and permanent tissue. The two types of plant tissues are meristematic and permanent. Then we have already studied about the meristematic tissue, which is made up of cells that are actively dividing, and it is found in different parts. We studied that meristematic tissues are classified into many categories on the basis of their location, on the basis of their function, on the basis of their origin. Then the second type of tissue is called permanent tissue, which have lost the power of division. Okay. Then we studied about, according to the position of meristematic tissue, where they are located in the plant body. Three different types of meristematic tissues are apical meristematic tissue, lateral meristematic tissue, and intercalary meristematic tissue. Then we studied about the permanent tissue. What are permanent tissue? Those tissues, plant tissue, which are made from meristematic tissue only, but once they become mature, grow old, and they lose their power of division, they attain permanent shape and size. Such plant tissues are called permanent tissues. So permanent tissues are further divided into two main types. What are they? They are simple tissue and complex tissue. Simple permanent tissue means those permanent tissues, that is those plant tissues which are made up of only one type of cell. Like one type of cell is present, as in case of parenchyma, collenchyma, sclerenchyma. So parenchyma, collenchyma, sclerenchyma are the three different types of simple permanent tissue which have different functions to do in the plant. Like parenchyma tissue, already have studied, is the one which is made up of cells that are oval or spherical, having equal diameter, that is called isodiametric, and they help in the storage of food. They are found in all parts of the plant. Like parenchyma is also present in the xylem, it is called xylem parenchyma. Parenchyma tissue is also present in the phloem, it is called phloem parenchyma tissue, and they may help in the process of photosynthesis, storage and secretion also. Then we studied about the second type of simple permanent tissue, that is collenchyma tissue, which is made up of cells which are thickened at the corners due to deposition of cellulose, hemicellulose and pectin. And collenchyma tissues can be spherical, oval or spherical. It may contain chloroplast. So there are certain modifications of parenchyma tissue also that we have studied. Those parenchyma tissue which contains air space inside them, particularly in aquatic plants, that help them to provide buoyancy or floating power, is called aerenchyma. The parenchyma tissue in which there is collenchyma tissue in which the parenchyma tissue which contains chloroplast is called chlorenchyma, and they will help in the process of photosynthesis. So what is the function of collenchyma if asked? In short, you can learn they provide mechanical support. How? Because of the presence of cellulose or pectic substances which are deposited at the corners of their cell walls. Okay? Now where they are found? They are found in the growing parts like young stem and the petiole of a leaf. The stalk of the leaf is called petiole. So here collenchyma tissue is present. It is also found in the growing tips. Then comes sclerenchyma. Sclerenchyma we studied. It is made up of cells which have very thick cell walls due to deposition of a substance called lignin, and it is of two types, that is fibers and sclerites, which are of different shape and they carry out different types of functions. They provide mechanical support. Then we have to studied about the complex permanent tissue. Complex permanent tissues are further divided into two types. What are they? Xylem and phloem. So first of all, you have to learn the definition of why these tissues are called complex tissue. The answer is because these tissues are made up of more than one type of cells. Like if you take the example of complex tissues, there are two types of complex tissue in plants. What are they? Xylem and phloem. Now this xylem is further made up of two types, four different types of elements. Phloem also made up of four different types of elements. And all the four elements of xylem are like xylem tracheids, xylem vessels, xylem parenchyma and xylem fibers. Okay, Fibers, fibers. So these are the four different cells which makes a xylem tissue. Xylem tracheids, vessels, parenchyma and fibers. And each of them carry out different types of functions. They have different shape and size. So the main conducting tissue is tracheids and vessels, which helps to form a pipe-like structure that help in the transportation of water and minerals from the roots to the upper parts of the plant. And one more important difference you can learn between xylem and phloem is inside the xylem, flow of water and minerals is unidirectional, means in a single direction. But in phloem, there is bidirectional movement of the organic food from one part of the plant to the other. Means in case of phloem, the glucose or the food can be transported in both directions, from the leaves, that is from the place where it is manufactured, to all other storage organs of the plant, as well as from all other organs of the plant to the leaves also it can come. Means there is movement of food in both the direction in the phloem tissue. And phloem tissue is also made up of 4 different types of cells. What are they? They are number 1, sheath tubes, number 2, companion cells, phloem parenchyma and phloem fibers. So one thing is common between them, that is xylem contains xylem parenchyma, phloem contains phloem parenchyma. Xylem contains xylem fibers, phloem contains the phloem fibers. So next one is, what are the 2 conditions of xylem, endarch and exarch? You should know the differences between them. Now, the protoxylem lies towards the center, and metaxylem, if it is present towards the periphery, like if I will draw a diagram like this is the center, the pith. If protoxylem is towards the center, I have drawn it very big. Wait, I will make it more clear. Okay, exarch and endarch. Xylem you should know. Okay. Okay, look at the diagram which I have drawn on the board. First I have written the classification of xylem. Xylem is of 2 types. What are they? Protoxylem and metaxylem. The meaning of the word proto means first, meta means the one which is formed later. So if the xylem is formed first, such a xylem is called protoxylem. The xylem which forms later on, that is called metaxylem. So 2 types of xylem is there. Now again 2 conditions of xylem is there which we need to learn. Endarch condition and exarch condition. So what happens is, in case of stem, when we study the TS, this is the TS of stem when we study, we find that in the center we have a region called pith, P-I-T-H, and it contains the vascular bundles, that is xylem and phloem is also there, conducting tissue is present. Now if the protoxylem is facing towards the center, that is towards the pith, and the metaxylem is facing towards the outside, this type of condition which is found in stem of the xylem is called endarched xylem. Got it? So what is endarched xylem? Endarched xylem is a condition of xylem in which protoxylem faces towards the center, that is towards the pith, and metaxylem is facing towards the periphery, means towards the outside. Now if you consider it in case of roots, it's just opposite. In roots, if we see the TS, the center is the pith is not, this one we find that if you study the xylem here, now which xylem will be outside? The protoxylem will be towards the periphery, the position of protoxylem. Whereas the metaxylem will be facing towards the center. So metaxylem is towards the center, and this type of condition of xylem is called exarch xylem. E-X-A-R-C-H. Exarch, endarch. E-X-A-R-C-H. Got it? So if it is asked, write the differences between exarch xylem and endarched xylem. Your answer should be, it is found in stem, endarched xylem. Exarch xylem is found in roots. In exarch xylem condition, protoxylem is towards the center, that is pith. Metaxylem is facing the periphery. Whereas in case of exarch xylem, protoxylem is facing the periphery, that is outside, and metaxylem is facing towards the center. Okay. Now next we have to study is phloem. Is made up of 4 different types of cells, I said. Again there are 2 terms, protophlloem and metaphlloem, similar to that of xylem. Like protoxylem is there, protophlloem is also there. The first form phloem is called protophlloem, and the phloem which develops later or which is formed later on is called metaphlloem. Then we studied about the tissue system. We studied in the interactive classes. What are the different types of tissue system? Epidermal tissue system, which consists of, just like our body is covered with the outermost layer of cells that is present in the skin. Similarly, the plants also have an outermost organ system or the tissue system which is called epidermal tissue system. And it is, in case of leaves, the epidermis is like interrupted by many tiny pores which are known as stomata, through which there is exchange of gases as well as removal of excess water in the form of, like during transpiration, in the form of water vapor. Now we find that in case of root, the outermost layer of root hair cell, which is the epidermal cell, it results, or the epidermal cell extends to form unicellular root hairs. So root hairs are unicellular, which is formed by the extension of epidermal, that is the outermost layer of cells present in root hairs. And what is the function of root hairs? It helps to absorb water and minerals from the soil. Then you know about the structure of stomata already I discussed, that stomata are of two different shapes. In case of monocot, the shape of stomata is just like a dumbbell. The guard cells are dumbbell shaped, like this. This is called a dumbbell shape. Whereas in case of dicot plants, the guard cells are just like a bean or kidney shape, like this shape is there. Okay, so this type of shape of stomata is present. The guard cells are kidney shape or bean shape in case of dicot plants. In monocot plants, they are shaped like a dumbbell. So here one more dumbbell you can draw. And this one is a stomatal pore is present in between them. Understood? So both the guard cells are dumbbell shaped, where in case of monocot plants. Both the guard cells are kidney shaped or bean shaped, where in case of dicot plants. And the gap or the pore which is present between them is called stomatal pore. The nearby cells which are present, they are called subsidiary cells. And you know what causes the opening and closing of stomata? It is the hardness or the turgidity of guard cells. Like when guard cells are filled with water, they become turgid, and there are several mechanisms that causes the opening and closing of stomata, like potassium ion concentration theory, glucose starch conversion theory is there. In the glucose starch conversion theory, it says that the chloroplasts which are present in the guard cells, these chloroplasts, they will trap sunlight and they will make food by photosynthesis. Okay, so as a result, there will be plenty of sugar molecules, less of water. So water molecules from the nearby cells, which are called subsidiary cells, from the nearby cells, the water molecules will diffuse into these guard cells. H2O will go. Why? Because plenty of glucose is there, less water is there. So when water will enter inside the guard cells will become turgid, that is, it will swell, and it will apply a pressure on the thick inner wall of the guard cell. This inner wall is thick, the outer wall is thin. So this wall is thick means due to turgid pressure, this pressure will be applied on the inner thick wall. That will result in the opening of stomatal pore. That happens in the daytime. At night there is no sunlight, no photosynthesis. So whatever glucose, excess of water is there in the guard cell, so those water molecules will go into the subsidiary cells. The guard cells will become flaccid, and it will result in the closing of stomatal pore. So that you have already studied, that regulation of opening and closing of stomata. Okay, now next we studied that one word is there, trichomes. What are they? The hairs which are present on the stem are called trichomes, and the root hairs, they are called unicellular root hairs only, or epigramma also you can say the name. Then we have to study about the ground tissue system. All the tissue systems, or all the tissues of the plant except epidermis and vascular bundles, it makes the ground tissue system. In case of leaves, the ground tissue system consists of mesophyll tissue, and mesophyll tissue is of two types, palisade and spongy. So already we have studied. Then we studied the different types of vascular bundles. What is vascular bundle? Vascular bundle consists of a xylem. It consists of this xylem, this one is phloem, and in between them there is cambium. Now if cambium is present, then such a vascular bundle in which cambium is present is, yesterday we had studied closed and open vascular bundles. Okay, so if the vascular bundles have no cambium present, they are called closed, But if cambium is present, it is called open vascular bundle, VV. VV means vascular bundle. So what is open vascular bundle? Such a vascular bundle in which cambium is present. Now if I draw a vascular bundle in which there is only xylem and phloem, but there is no cambium between them, such type of vascular bundle is called closed vascular bundle. Okay. So today in the introductory class also I will conduct. Then there we can start with some more portions like the diagrams and all. I will show you and explain that diagram. Okay. Thank you.